Welcome to the deep dive. Today, we're pulling back the curtain on one of the body's most incredible silent conductors, the endocrine system. Have you ever wondered how your body manages to orchestrate countless functions, from responding to a sudden fright to maintaining your metabolism, all with such incredible precision?
It's quite amazing when you think about it.
It really is. It's a symphony of communication. And while you might immediately think of the nervous system, that rapid wired network, there's another equally vital, more chemical communication system working tirelessly behind the scenes.
That's right. And for this deep dive, our mission is to really unravel the dense, highly detailed material from Chapter 47, Organization of Endocrine Control in Boron and Boulpaep's Medical Physiology.
A classic text.
Absolutely. Our goal is to transform this intricate physiological information into clear conversational explanations that are still academically accurate, and crucially, connect these complex concepts to real-world clinical relevance.
So you can actually see how it applies.
Exactly. We're going to build your understanding step by step. Start with the big picture before diving into the fascinating details.
Okay, so the big picture. We've got these two major communication systems. You mentioned the nervous system, lightning fast, great for reflexes like pulling your hand off a hot stove.
Yeah.
Quick integration.
Right, wired communication. And then you have the endocrine system. This system integrates organ function, not with quick electrical impulses, but through chemical messengers.
Hormones.
Hormones, exactly. These powerful chemicals are secreted from specialized endocrine tissues or glands directly into the extracellular fluid. From there, they typically journey through your bloodstream, carried sometimes quite far, to distant target tissues. They orchestrate this much broader, often slower, but incredibly profound symphony of changes throughout your entire body.
Let's zero in on these hormones then, these chemical signals. What makes them work so well?
Well, the secret really lies in their recognition by incredibly specific, high-affinity receptors. Think of it like a perfectly matched lock and key. These receptors can be right on the surface of the target cell, or sometimes, maybe surprisingly, even inside the cell, sitting in the cytosol, or maybe even right in the nucleus.
Inside the cell. Wow.
Yeah, and the accuracy of this recognition is truly remarkable. Hormones can circulate at incredibly low concentrations. We're talking 10 to the minus 9, even 10 to the minus 12 molar. That's minuscule.
Like finding a needle in a haystack?
Pretty much, yeah. Yet, your cells detect and respond precisely. Once a hormone finds its specific receptor, it kicks off a process called signal transduction.
Okay, signal transduction, that's converting the message.
Exactly, converting the hormone's message into a cellular action. And the responses can vary wildly in timing. Some are almost immediate, like epinephrine ramping up your heart rate in seconds, or glucagon quickly breaking down liver glycogen.
Right, fast responses.
But other actions take much longer hours or even days. Think about aldosterone's role in salt retention by the kidney, or a growth hormone promoting protein synthesis and growth over time. It really highlights the diverse timescales these messengers operate on.
So, besides just speed, how does the body make sure these hormones hit the right targets? Are there different ways they send signals?
Absolutely. The body uses three main strategies, each with its own sort of advantage. First, there's what we call classic endocrine signaling. This is probably the one most people think of.
The textbook example.
Right. A hormone is secreted by a gland, travels a long distance through your systemic circulation, the bloodstream, and then reaches a distant target tissue, like sending an official letter through the mail.
Goes everywhere, but only read by the recipient.
Precisely. Then you have paracrine action. Here, hormones regulate nearby cells without entering the general circulation. They just act locally in the extracellular fluid, affecting their immediate neighbors.
So more like a local memo or a quick text to someone nearby.
Exactly. Good analogy. It's local and direct. And finally, there's autocrine regulation. This one's kind of neat. A chemical binds to receptors on the very same cell that secreted it.
So the cell is talking to itself.
Essentially, yeah, it's affecting its own function, like an internal feedback loop to fine-tune its own activity. And the really amazing thing is it's the combination, the summation of all these actions, classic endocrine, paracrine, and autocrine, that creates these incredibly complex and refined regulatory systems in your body.
Okay, so we have the messengers and the delivery methods. Where do they actually come from? We talk about the classic endocrine glands.
Right, the traditional list. You've got the pituitary, thyroid, parathyroids, testes, ovaries, the adrenal glands, both cortex and medulla, and the endocrine part of the pancreas. Those are the 7 classics.
But it's not just them, is it?
No, definitely not. It's crucial to understand that many other tissues, ones we don't typically label as endocrine glands, also produce hormones. Parts of the central nervous system, especially the hypothalamus.
Which sits right above the pituitary.
Exactly. Also the gastrointestinal tract, the liver, heart, even the kidneys. They all chip in, contributing to this chemical communication network.
And this leads to something really important clinically, doesn't it? This idea of non-endocrine tissues making hormones.
Oh, absolutely. It's a fascinating and clinically vital phenomenon called neoplastic hormone production. Sometimes certain types of cancers originating in tissues that normally don't make a particular hormone can start producing it.
Like a mix-up in production.
Kind of. Think of it like the body's alarm system getting cross-wired. For example, some lung cancers might start churning out EVP, arginine vasopressin.
The water balance hormone.
Right. And that can lead to hyponatremia, dangerously low blood sodium. Other lung tumors might make ACTH, causing Cushing syndrome, or maybe a PTH-related peptide, mimicking parathyroid hormone and causing hypercalcemia, high calcium levels.
Wow. And the key thing is,
The really critical point is that these symptoms, these paraneoplastic syndromes, can actually show up before there's any other sign or suspicion of the cancer itself.
so they can be the first clue.
Absolutely, making them crucial diagnostic leads for clinicians. It really underscores how understanding basic physiology helps in diagnosis. Interestingly, most of these ectopic hormones produced by tumors tend to be peptide hormones.
Speaking of peptides, you mentioned the lines can blur. What about neuropeptides?
Good point. They really bridge the gap. Take somatostatin, for example. It's a peptide made by delta cells in the pancreas. There, it acts locally, paracrine action, to regulate insulin and glucagon secretion.
Okay, local regulation in the pancreas.
But somatostatin is also made by neurons in the hypothalamus. These nerve endings release it into that special pituitary portal blood system. It travels just a short distance to the anterior pituitary and inhibits growth hormone secretion.
So the same molecule acts locally in the pancreas and as a neurohormone affecting the pituitary.
Exactly. It perfectly illustrates how interconnected the nervous and endocrine systems are.
Okay, let's shift gears slightly. What are hormones actually made of, chemically speaking?
Right. Most mammalian hormones fall into one of three main chemical groups. First, a really large group, the peptide hormones. These include lots of familiar ones: insulin, growth hormone, GH, TSH, ACTH, LH, FSH, prolactin, PRL, parathyroid hormone, PTH, a whole bunch, made by lots of different endocrine tissues.
Okay, peptides are common. What else?
Then you have the amino acid-derived hormones. Many of these are built from the amino acid tyrosine, think epinephrine, norepinephrine, dopamine, and also your thyroid hormones, T3 and T4. Others, like serotonin, come from tryptophan.
Amino acid building blocks. Got it. And the third group?
The third group are the steroid hormones. Fascinating group. These are all synthesized from cholesterol. This includes hormones like cortisol, aldosterone, estradiol, progesterone, testosterone.
All from cholesterol.
Yep. Now, while peptide hormones are made all over, the catecholamines, like epinephrine and the steroids, require specialized enzymatic steps. Only certain tissues have the machinery. And thyroid hormone synthesis, that's pretty much restricted to the thyroid gland. These specialized pathways give specific tissues control over producing these potent signals.
So once they're made and secreted, how do they travel? You mentioned the bloodstream. Do they just float freely?
Some do, yes. Many circulate freely. But others, especially thyroid hormones and steroid hormones, and also IGF-1, IGF-2, and growth hormone, form complexes with circulating binding proteins.
Binding proteins, what do they do?
They serve a few really important functions. First, they act as a reservoir, kind of buffering the concentration, minimizing rapid fluctuation.
Smoothing things out.
Exactly. And second, they significantly extend the hormone's half-life in the blood. Take T4, thyroid hormone. Over 99.9% of it is bound to proteins. This gives it a half-life of about 7 or 8 days. If it were free, its half-life would be measured in minutes.
Huge difference.
Massive. Generally, hormones that are mostly bound tend to have longer-term actions, while the free, unbound hormones often play more immediate short-term roles.
And this bound versus free concept is clinically important, right?
Absolutely critical. You'll definitely encounter this. Think about pregnancy. The liver ramps up production of T4 binding globulin. So if you measure total T4 in a pregnant patient's blood, it will be high because there's more binding proteins soaking it up.
But that doesn't mean she's hyperthyroid.
Exactly. The physiologically active part, the free T4, the bit that can actually enter cells and do its job, generally remains unchanged. Why? Because the pituitary gland senses the free T4 level and adjusts TSH secretion accordingly to keep it stable. Understanding the difference between total and free hormone levels is fundamental for interpreting lab results correctly in many endocrine situations.
Okay, that's a really key point. So how do we measure these tiny amounts of hormones? You mentioned the concentrations are incredibly low.
Right. And the ability to do that accurately really revolutionized endocrinology. The story starts back in the late 1950s with Solomon Bersin and Rosalind Yalow. They made a truly groundbreaking discovery. They found that patients who received insulin injections actually developed antibodies against it.
Antibodies against insulin.
Right.
But insulin is made by the body.
Exactly. That was the first huge implication. It proved that the body's immune system can react to its own endogenous compounds. This flew in the face of the thinking at the time and really laid the groundwork for understanding autoimmune diseases, things like type 1 diabetes, autoimmune thyroid disease, Graves' disease.
Where the body attacks itself.
Precisely.
Yeah.
Before this, the dogma was largely that the immune system didn't react to self. So, massive paradigm shift.
Okay, that's one implication. What was the second?
The second, and crucial for measurement, was their realization that these antibodies had such a high affinity, such a strong attraction for insulin. They figured they could use these antibodies to measure the tiny amounts of insulin in blood serum. This led directly to the development of the radioimmunoassay, or RIA.
Radioimmunoassay. How does that work conceptually?
Okay, picture this vocally. Imagine you have a test tube with a fixed known amount of a very specific antibody that binds to the hormone you want to measure, let's say, insulin.
Got it. Antibody ready.
Now, you add a known amount of insulin that's been tagged with a radioactive label. This labeled insulin binds to the antibody, filling up the available spots.
Okay, labeled hormone bound.
Now comes the clever bit. You add your sample, say, serum from a patient, which contains an unknown amount of unlabeled insulin. This unlabeled natural insulin competes with the radioactive insulin for those binding spots on the antibody.
Competition.
Exactly. The more unlabeled insulin there is in the patient's sample, the more it will displace or kick off the radioactive insulin from the antibody.
So, less radioactivity bound to the antibody means more hormone in the sample.
Precisely. You measure the amount of radioactivity that remains bound, or sometimes the amount that gets displaced. By comparing this to a standard curve you create using known amounts of unlabeled hormone, you can accurately figure out how much hormone was in the patient's original sample.
That's incredibly clever.
It really is.
Yeah.
The high specificity and affinity of these antibodies allow for detecting hormones at those pico or nanomolar concentrations. RIA and its modern descendants, using non-radioactive labels like chemiluminescence, totally transformed endocrine research and diagnostics. It's why Rosalind Yalow won the Nobel Prize.
Amazing story. Okay, so we know how hormones signal and how we measure them. How do they work together? Do they cooperate?
Often, yes. Many complex functions require what we call complementary action, where multiple hormones pitch in for a common goal. Think about strenuous exercise.
Yeah.
To manage that stress and mobilize energy, you need epinephrine, cortisol, and glucagon all working together. If you're deficient in any one of those, your exercise capacity plummets, and you could even get dangerously low blood sugar, hypoglycemia.
So it's a team effort.
Definitely. Another example is normal growth. It requires growth hormone, GH, insulin-like growth factor 1, IGF-1, thyroid hormone, and sex steroids all playing their part. If you're missing adequate GH, IGF-1, or thyroid hormone, the result can be dwarfism. Shows how critical that teamwork is.
But sometimes they work against each other too, right?
Absolutely. That's antagonistic action. Hormones with opposing effects. In these cases, the overall result on a target organ depends entirely on the balance between these opposing forces.
Like a tug of war.
Kind of, yeah. The classic example is blood glucose control with insulin and glucagon. Insulin acts to lower blood glucose. It tells the liver to stop making glucose and helps muscle and fat take it up.
Okay, insulin brings glucose down.
Right. Glucagon does the opposite. It raises blood glucose by stimulating the liver to break down stored glycogen, glycogenolysis, and make new glucose, gluconeogenesis. This constant push and pull, this delicate balance, provides really fine-tuned control over glucose levels and many other cellular functions.
That makes sense, and all this regulation relies on
Feedback control. This is the heart of endocrine regulation. Any good regulatory system needs to sense when to increase or decrease its activity, right?
Sure. Needs to know when the job is done or needs doing.
Exactly. In the endocrine system, the hormone-secreting cell often acts as the sensor. It monitors a specific regulated variable, maybe blood glucose, maybe calcium levels, maybe even the concentration of another hormone.
Okay.
Based on what it senses, it adjusts its own hormone secretion. That hormone then acts on target tissues, changing the variable, and that change is then sensed back by the original cell, completing the loop.
Closing the loop, like the thermostat in your house.
Perfect analogy. A simple biological example is insulin secretion by pancreatic beta cells. High plasma glucose after a meal. Beta cells sense it, secrete insulin. Insulin acts on liver and muscle, lowers glucose. Lowered glucose is sensed by the beta cell, which then reduces insulin secretion. Beautifully simple, incredibly effective.
But it can get more complicated than that, adding more layers.
Oh, definitely. We often see hierarchical control, which frequently involves the central nervous system, adding levels of command. The CRH-ACTH cortisol axis is a prime example.
Okay, walk us through that one, stress response.
Right. Imagine you're under stress, say, a serious infection or injury. Your cerebral cortex signals the hypothalamus to release corticotropin-releasing hormone, CRH.
Okay, step 1, CRH from hypothalamus.
Step 2. CRH travels through that special pituitary portal system, just a short hop to the anterior pituitary. There, it stimulates specific cells to release adrenocorticotropic hormone, ACTH.
Step 2, ACTH from anterior pituitary.
Step 3. ACTH travels through the main circulation to the adrenal cortex, the outer part of your adrenal gland, and tells it to synthesize and release cortisol.
Step 3, cortisol from adrenal cortex. And cortisol is the stress hormone that does the work.
It is. It acts on many tissues to help the body cope with the stress. But here's the elegant feedback part.
Closing the loop again.
Exactly. Cortisol itself then feeds back to inhibit CRH release from the hypothalamus, and it also makes the pituitary less sensitive to CRH, reducing ACTH release.
So it shuts off its own production upstream, prevents runaway cortisol levels.
Precisely. It's a self-regulating system. This brings us nicely to the pituitary gland, the hypophysis. Located right at the base of the brain, under the hypothalamus, it's a real master gland, bridging neural and endocrine control. And it has two distinct parts, or lobes.
Anterior, posterior.
Right. The anterior pituitary is true glandular tissue. It makes and secretes 6 key peptide hormones. Growth hormone, GH. Thyroid-stimulating hormone, TSH. ACTH, which we just met. Luteinizing hormone, LH. Follicle-stimulating hormone, FSH. And prolactin, PRL.
That's a busy gland.
It is. And crucially, the release of these hormones is controlled by those hypothalamic releasing hormones, or in some cases, inhibiting hormones like GHRH, TRH, CRH, GnRH, and dopamine. They travel down that portal system to tell the specific anterior pituitary cells what to do.
And 4 of those pituitary hormones, TSH, ACTH, LH, FSH, they then go on to stimulate other endocrine glands, right? Like a chain of command.
Exactly. They are often called trophic hormones because they stimulate other glands. TSH goes to the thyroid, ACTH to the adrenal cortex, LH and FSH to the gonads.
Okay. What about prolactin, PRL? You said it was unique.
Yeah, prolactin is a bit different. Its secretion is normally inhibited by dopamine coming down from the hypothalamus. There isn't a classic downstream hormone that feeds back to shut it off. Instead, things like breast stimulation during nursing send neural signals up to the hypothalamus to decrease dopamine release.
Removing the brakes.
Exactly. Taking the foot off the brake allows prolactin secretion, enabling milk production. It's a neat example of direct neural control over an endocrine hormone.
Okay, that's the anterior pituitary. What about the posterior?
The posterior pituitary is fundamentally different. It's not actually glandular tissue. It's neural tissue, a direct extension of the brain, specifically the hypothalamus.
An extension of the brain.
Yes. It contains the long axons and nerve terminals of large neurons whose cell bodies are located up in the hypothalamus. These hypothalamic neurons actually make the hormones arginine vasopressin, AVP, also called ADH, and oxytocin.
So the hormones are made in the hypothalamus.
Correct. They're synthesized up there, then transported down the axons and stored in the nerve terminals within the posterior pituitary, waiting for the signal to be released directly into the bloodstream.
And what do AVP and oxytocin do?
AVP, or antidiuretic hormone, acts mainly on the collecting ducts in your kidneys to increase water reabsorption. Hugely important for maintaining water balance.
Prevents dehydration.
Essentially, yes. Oxytocin is best known for stimulating smooth uterine contractions during childbirth and milk ejection from mammary glands during suckling.
Important roles. Is there a clinical link here too?
Yes, definitely. If there are defects in making or processing the AVP precursor protein back in the hypothalamus, it can lead to central diabetes insipidus. This isn't related to blood sugar diabetes, it's about water. Patients can't reabsorb water properly in their kidneys, so they produce huge volumes of very dilute urine and have excessive thirst.
Okay, fascinating distinction. Now let's dive even deeper. How do these hormones actually work once they reach a target cell? Let's start with the peptide hormones.
Right. Peptide hormones, we know they're made via the usual protein synthesis route, rough ER, Golgi, stored in vesicles, released by exocytosis, often on demand.
Okay.
The key thing is, once they're secreted, they generally circulate freely, and because they're usually longer-soluble and often large, they can't easily slip through the cell membrane, so they bind to receptors located on the surface of their target cells.
On the outside, like ringing the doorbell.
Good analogy. The receptor is the doorbell. When the hormone, the finger, pushes it, it triggers a chain reaction inside the house. That's the signal transduction we mentioned earlier.
And how does that internal chain reaction work? What are the mechanisms?
There are several major pathways. A very common one involves G-protein-coupled receptors. Many hormones, like PTH or glucagon or TSH, bind to these receptors. Binding activates a G protein on the inner surface of the membrane.
A molecular switch.
Exactly. This activated G protein then often turns on an enzyme called adenyl cyclase. Adenyl cyclase converts ATP into cyclic AMP or King MP.
KMP, I've heard of that, a second messenger.
Precisely. CMP is a classic second messenger. It then typically activates another enzyme called protein kinase A, pKa. pKa then goes around phosphorylating, adding phosphate groups to various cellular proteins and enzymes, changing their activity and causing the cell's ultimate response.
So hormone receptor models G protein, adenyl cyclase, Grecinale, Pangas, pKa, cellular response.
Right.
Quite a cascade.
It is, and understanding this is key clinically. For example, pseudohypoparathyroidism is a condition where patients have plenty of PTH, but their cells don't respond properly because there's a defect in the G protein, specifically the G-alpha subunit, that's supposed to link the PTH receptor to adenyl cyclase. The signal gets stuck.
So the doorbell rings, but the message doesn't get passed on.
Exactly. Another major G protein pathway involves phospholipase, PLC. Hormones like AVP, acting on V1 receptors, or TRH, bind their receptors, activate a different G protein, GQ, which then activates PLC. PLC chops up a membrane lipid into two second messengers, IP3 and DA.
Two messengers from one reaction.
Yep. IP3 typically triggers the release of calcium from internal stores, and DA activates protein kinase C, PKC. Both calcium and PKC activation lead to changes in cell function. There are other pathways, too, involving things like phospholipase A2 or guanylyl cyclase receptors, but CMP and IP3-DAG are really big ones for peptide hormones.
What about receptors that are enzymes themselves, like tyrosine kinases?
Right. Some receptors, notably for insulin and the insulin-like growth factors, IGFs, are receptor tyrosine kinases. The part of the receptor inside the cell actually has enzyme activity. When the hormone binds on the outside, it switches on this internal kinase activity.
So the receptor itself does the phosphorylating.
Yes. It phosphorylates itself and other target proteins on tyrosine residues, initiating a signaling cascade. Other receptors, like the one for growth hormone, don't have intrinsic kinase activity but are tightly associated with separate cytoplasmic tyrosine kinases, often from the JAK family. Hormone binding brings the receptor parts together, activating these associated JAK kinases, which then phosphorylate targets, including STAT proteins, leading to changes in gene expression.
Okay, so peptides work from the outside via surface receptors and second messengers or kinase cascades. What about the amine hormones? Epinephrine, norepinephrine, thyroid hormones?
Well, the catecholamines epinephrine, norepinephrine, dopamine act much like peptide hormones. They circulate freely and bind to specific cell surface receptors, like the various adrenergic receptors, alpha-1, alpha-2, beta-1, beta-2, beta-3. These are typically G-protein-coupled receptors.
And the effect depends on which receptor is present.
Absolutely critical. Epinephrine binding to a beta-1 receptor in the heart speeds it up, but binding to a beta-2 receptor in the airways relaxes smooth muscle. Same hormone, different receptor, different effect.
Makes sense. What about serotonin?
Serotonin also acts via surface receptors. Clinically, it becomes really interesting with carcinoid tumors. These tumors can arise, often from the gut, and secrete massive amounts of serotonin. This leads to the dramatic carcinoid syndrome characterized by episodes of intense skin flushing, severe diarrhea, sometimes wheezing or bronchospasm. These symptoms can be a vital clue to diagnosing the underlying tumor.
Wow. Okay, so peptides and catecholamines mostly work from the cell surface. That leaves steroids and thyroid hormones. You said they were different.
Very different. Steroid hormones, like cortisol, aldosterone, estrogen, testosterone, are derived from cholesterol, remember? They're lipid-soluble.
So they can pass through the cell membrane.
Exactly. They diffuse right across the plasma membrane into the cell. And importantly, they are not stored in vesicles. Their synthesis from cholesterol and immediate secretion are tightly linked and regulated.
Okay, they're inside the cell now. What happens?
Once inside, they bind with high affinity to specific receptor proteins located either in the cytosol or, more often, directly in the nucleus.
So the receptor is inside, not on the surface.
Correct. This hormone receptor complex then gets activated and binds to specific sequences on the DNA called hormone response elements, or specifically for steroids, steroid response elements, SREs. These are usually located near the genes that the hormone regulates.
Binding to DNA, so they're controlling genes.
Precisely. Binding of the hormone receptor complex directly regulates gene transcription, turning genes on or off, or modulating how actively they're transcribed into messenger RNA. This leads to changes in the synthesis of specific proteins, which ultimately causes the cellular response. Because this involves making new proteins, these effects typically take longer to develop, hours to days.
So they're fundamentally changing cell function by altering protein production. That's a powerful mechanism.
Incredibly powerful, and this has direct clinical applications. Think about quantitating steroid receptors in cancer, especially breast cancer. Doctors can take a biopsy of a breast tumor and measure the levels of estrogen receptors, ER, and progesterone receptors, PR, in the cancer cells, often using methods similar in principle to the amino acids we discussed.
Why measure those receptors?
Because if a breast cancer cell has high levels of ER or PR, it tells us two things. Generally, it correlates with a somewhat better prognosis, and more importantly, it means the cancer's growth is likely driven by estrogen. Therefore, treatments that block estrogen action, like the drug tamoxifen, are much more likely to be effective. It's a cornerstone of personalized cancer therapy, using receptor biology to guide treatment.
That's a fantastic example of bench-to-bedside translation. What about thyroid hormones, T3 and T4? Are they similar?
They share similarities with steroids. They're synthesized uniquely within thyroid follicles. They also readily enter target cells and bind to intracellular receptors, mostly in the nucleus, although some binding occurs in the cytosol too. T3 is generally considered the more active form at the receptor level.
And they also regulate genes.
Yes. The T3 receptor complex binds to thyroid hormone response elements on DNA and regulates the transcription of target genes. Thyroid hormones play a huge role in setting the overall metabolic rate of the body, affecting growth, development, and the function of almost every organ system. That's why thyroid disorders, hyperthyroidism or hypothyroidism, cause such widespread symptoms.
So steroids and thyroid hormones work mainly through intracellular receptors and gene regulation. Any exceptions?
Actually, yes. This is a relatively newer area of understanding, but it's becoming clear that both steroid and thyroid hormones can also exert rapid non-genomic action.
Non-genomic?
Yeah.
Meaning not involving changes in gene expression.
Exactly. These effects happen within minutes, which is far too quick to be explained by synthesizing new proteins. It seems these hormones can sometimes bind to proteins in the cytosol, or perhaps even receptors on the cell membrane, and directly modulate their activity or trigger faster signaling pathways.
Can you give an example?
Sure. There's evidence that the estrogen receptor, when bound by estradiol, can directly interact with and activate a signaling molecule called PI3K, phosphatidylinositol 3 kinase in the cytosol. This can rapidly influence things like vascular function, completely independent of gene transcription. So the classic genomic pathway is dominant, but these faster non-genomic actions add another layer of complexity.
Wow. So the picture is always evolving. We've covered a huge amount today.
We really have.
From the basic organization, the nervous versus endocrine systems, to the different types of hormones and how they signal classic paracrine, autocrine. We looked at where they come from, including those important clinical links with neoplastic production.
Right. Then their chemical nature, peptides, amines, steroids, how they travel boundary-free, and how we measure them with immunoassays, that fascinating RIA story.
And then the regulation, complementary, and antagonistic actions, the crucial feedback loops, the hierarchical control involving the pituitary and hypothalamus.
Focusing on the anterior pituitary's 6 hormones and the posterior pituitary's 2 hypothalamic hormones, AVP and oxytocin.
And finally, diving right down to the cellular mechanism's surface receptors with second messengers and kinases for peptides and catecholamines versus the intracellular receptors and gene regulation for steroids and thyroid hormones, plus those emerging non-genomic effects.
It's a lot, definitely complex material. But breaking it down like this step by step helps build a real framework, I hope. You're not just memorizing isolated facts, but hopefully seeing how it all connects from basic physiology to clinical practice.
Absolutely. It's about understanding the why and how. As you go about your day, maybe just take a moment to consider that silent chemical orchestra playing within you, constantly adjusting, constantly regulating. It makes you wonder what other unseen systems might be orchestrating our existence in ways we're only just beginning to grasp.
A profound thought.
Remember, you're part of the deep dive family, and you're absolutely capable of mastering this material. Keep diving deep.